Hello, and welcome to PHEN's webinar on this beautiful day of June 30th. I'm glad to see that you all have joined us today. My name is Thomas Farrington. I am the president and founder of PHEN, the Prostate Health Education Network, and we're excited with the webinar that we have today. I am a 21-year prostate cancer survivor, and one of the things that I've seen during my 21 years as a survivor and the time that I have led PHEN since 2003 is just how exciting new developments take place within prostate cancer. We have some brilliant minds in the area of research. We have some brilliant minds developing clinical trials, and we see so many new therapies that are approved for patients. And, you know, again, as a patient, we get excited about this. And tonight we're going to talk about a new area. We're going to talk about PSMA. I'm not going to tell you what it is because I'm sure some of you know and some of you don't, but that's part of this webinar, to tell you what it is and what it means for patients. So again, I am so happy to have you with us today. Those of you that have joined us on Zoom, those of you that have joined us on Facebook, we're very, very excited. Now, we have two great presenters for this evening, and Dr. Keith Crawford will introduce our guest speakers to you. What I'm going to do now is to introduce Dr. Keith Crawford. But before I do that, let me outline how this webinar will be conducted. We will have presentations by both our guest speakers, and you are invited during either of these presentations to submit your questions on the Q&A section of Zoom. You look down, you'll see Q&A. At any time, you can submit a question to Q&A. Those questions will be answered, however, after both our presenters have completed their remarks, and then we will read those questions and answer them for you. So then at the conclusion of the panel discussion, we will also have a poll, and the poll will be anonymous. So I want you to take the time to answer the questions that are polled. We like to determine how well we've done in terms of bringing you information and educating you, and you know, preparing you for, again, the exciting future that's taking place within the prostate cancer space. So at this time, let me introduce Dr. Keith Crawford. Dr. Crawford joined PHEN a couple of years ago as our director of clinical trials and patient education. He has over 20 years in the life sciences area, and he completed his graduate and postgraduate training at Harvard Medical School, where he developed competence in the area of genomics, proteomics, immunology, microbiology, infectious disease, and regenerative medicine. Dr. Crawford received his undergraduate degree at Prairie View College, A&M College in Texas. He's very proud of that, and I know he would want me to mention Prairie View. So we mentioned Prairie View. He's a Texan, and but we accept him here in Boston. So he's a transplanted Texan to Boston. So at this time, I want to bring you Dr. Keith Crawford. Dr. Crawford.
Thank you, Tom. It's after 6, so good evening, everyone. And thank you again for joining us on this June 30th webcast. Now, I want to introduce our guest speakers. And it's important because they're going to I'm going to do an earlier presentation and then have them tie in the technology into the clinical trials that they will discuss. As you know, the title is PSMA. What is it and why it matters for the prostate cancer patients? And our panelists are Dr. Michael Morris, and he is a medical oncologist from MSKCC at Sidney Kimmel Center for Prostate Cancer. And then we also have this evening Dr. Michael Ronan, and he's the global affairs lead at Amgen. Now, I want to go back because this is a very exciting time in the prostate cancer care continuum as relates to also clinical trials. So I'm going to share my screen with you right now. And what we like to do at PHEN is I'm going to tell you what I'm going to tell you. I'm going to tell you, and I'm going to tell you what I told you. And what you'll hear is some redundancy, but we want everyone to understand how important it is and what the new findings that we're going to talk about, our guests or panelists are going to talk about later on. So the agenda, what is prostate specific antigen, PSA? So everyone's heard about that. But what is prostate specific membrane antigen? And this is PSMA. And I highlight membrane because I want people to be focused on the fact that we're talking about on the cell surface. What is the future role of PSMA in prostate cancer diagnosis and treatment? And then we'll have discussions, presentations by our guests. Dr. Morris can shed shed some light on the VISION trial, and Dr. Groening can shed some light on Amgen 160. What we also like to do is this is our opportunity, what we provide a certain level of education for the survivors and for their loved ones and everyone in the audience. So we always want to give a vocabulary list and to kind of bring everybody up to speed, because you'll hear these terms and words throughout the presentation. And the first is a biomarker, a characteristic of a cancer that can be measured and provide more information about your cancer. Prostate-specific antigen, you've heard about the term PSA. Now, this is a protein produced by normal as well as cancerous cells of the prostate gland. The PSA test measures the level of PSA in a man's blood. Prostate-specific membrane antigen, PSMA, is expressed upwards to 1,000 times in prostate cancer. And its degree of expression seems to correlate with the tumor differentiation and the presence of metastatic disease. You'll start to hear more and more on our presentation, but in the future, concepts or terms called theranostic. And theranostic combines the diagnostic tool that helps to define the right therapeutic tool for a specific disease. So they're just combining theranostics together as one word, kind of gives you. And it's something else that you'll start to hear more and more is this concept called personalized medicine, PM. And this is the right term, the right treatment for the right patient at the right time and the right dose. So personalized medicine shifts from targeting the disease to targeting the specific tumor of the patient. Now, PSA. We know that the PSA is a test. Generally, this is the first step in screening for prostate cancer, which is PSA. PSA is a protein produced by normal as well as cancerous cells of the prostate. PSA test was approved by the FDA to monitor prostate cancer growth, but is also used for early detection. PSA requires that your blood be drawn and sent to a laboratory for measurements. The PSA is not elevated only in men with prostate cancer. You can find it in men that have no cancerous background or cancerous tissue, trauma, sexual intercourse, even benign growth of the prostate. You'll have an elevated PSA. Now, prostate-specific membrane antigen. Again, I highlighted. So I want people to remember that when we're talking about PSMA, we're talking about something that's stuck to or found on the cell of the cancer cell. PSMA is just like PSA, is a novel prostate cancer biomarker. And so to define the biomarker, again, a biomarker is a characteristic of a cancer that can be measured or provide more information about the cancer. Now, PSMA more than 80% of the men with prostate cancer express PSA. So again, great biomarker, novel biomarker, and 80% of the men who have prostate cancer express PSMA. And again, I'm going to show you, this is just to get an idea of visualize. This is a very high magnification, scanning electron microscopy, but just think about it as a high magnification of cells on a slide. These are cancerous cells. And it gives you an idea of the PSMA protein stuck on the surface of a prostate cancer cell. In metastatic prostate cancer, if your prostate cancer cells express a large amount of PSMA, the cancer is considered PSMA positive. So let's summarize. Is PSMA different than PSA? Yes, it's similar. There's some overlapping areas. So let's walk down these areas. Both of them produced by the prostate gland. So both. Found on the surface? Nope, only PSMA. Found in the blood? Generally speaking, PSA. Important prostate cancer biomarkers, both of them are. Detected by the blood test, PSA. Detected with a PET or positronic emission or imaging technology, PSMA. And I want to take it a little bit further as we move from just the concept of just prostate cancer in general. And what can PSMA tell you about advanced prostate cancer? And this is where this is very important because we're getting a lot of new data from patients that are metastatic castration resistant, more advanced disease of prostate cancer. So PSMA and PSA are distinct biomarkers. So I've said that before, which can tell you different things about your prostate cancer diagnosis. PSMA can be used to show you whether prostate cancer in your body, where prostate cancer in your body, and if it is progressing. PSMA can give you or your healthcare provider more information about your disease status, which can help you during your decision-making process. So, where is it in the body? Is it progressing? The more information that you have, the better you can sit down with your clinician to map out your treatment options. And so I like visuals that kind of drive it home. So we're looking at a PSMA targeted theranostic design. And you have the cell itself. So for example, this is your prostate cancer cell. Then you have your target. In this case, it's your PSMA protein, again, found on the surface. Then you have a targeting agent. Now, a targeting agent is either a small molecule or a biologic like an antibody. And so we're using, in the case of the clinical trials that we'll be discussing after this segment, we'll be talking about an antibody-like or an antibody that serves as a targeting agent. Now, what's interesting is they take it a step further. Now they have a linker component that links your target agent to your payload. Now your payload can carry either a radioactive molecule that you can use for imaging or a radioactive molecule that can serve as a therapy or another molecule or a cell that can be attached to the payload that can bring a T cell or an immune cell in close proximity to the cancer cell and cause localized killing. Now, this is kind of an initial concept because we always talk about this prostate cancer continuum of care. And we want people to recognize that, stay positive, because more and more information is coming down the pipeline that can better help or help guide you on your journey. But what's interesting about PSMA is that you can use it in the diagnosis, making the diagnosis. Now with an imaging, PET scan, or a CT, and the prognosis. So PSMA levels in tumors have been negatively correlated with survival outcomes. So the higher the levels, the poorer the outcomes. So now you a prognostic indicator. And lastly, clinical management. There's reports that out of Australia and groups here in the United States demonstrate that comparing PSMA PET or CT with conventional imaging could lead to management changes in up to 60% of the patients in their studies. So we're talking about the future, the present, and the future of PSMA targeting. It's here, and we want everyone to get excited about this. Here this evening, there may be other future applications, but we're going to talk about more along the line of biologics or antibody molecules, like molecules that can bring a payload, whether it's radioactive or an immune cell, to that site and to concentrate and kill the cancer. Okay, so Dr. Morris, I wanted to, you've had some exciting news about the VISION trial. And I think that everyone has either read about it or heard about it. And I wanted you to have an opportunity to kind of tell us about the VISION trial and why it's important for the prostate cancer patients to really be excited about this new breakthrough and your findings.
Sure thing. So first, I do want to thank you, Keith, and thank PHEN for having me and inviting me to present, discuss the VISION trial and the whole field of theranostics. And I really enjoyed your introduction to the world of PSMA and PSMA therapeutics and diagnostics. Really covered things quite well. I have just one or two slides to show to discuss a little bit about, sort of as an introduction to VISION. So to answer your question as to why it's important and why we think that it'll change the landscape of treatment for prostate cancer. So if you have no objection, I'm just going to share my screen for a second, if that's okay by you.
That's okay. I'm happy to share it with you.
Okay, very good. So let me just share my screen here, and I'll turn that into a full screen view. And let me just swap this. All right. Can you see that diagram well?
Yes.
Great. I'm just going to show in cartoon form first before I show this slide, which summarizes exactly what you were just talking about, but just shows it shows the molecule that we're talking about right now, which is PSMA. And this is what it looks like in a cartoon form. And just as you were talking about it being having an extracellular component, a component that's outside the cell, that's this part of the cartoon. This here is the cell membrane. That's sort of like the covering of the cell that keeps the contents of the prostate cancer cell separate from its environment. This is the interior of the cell. And you can see that the outside portion of PSMA is pretty big and makes for a pretty good target to link up to and bring a payload, as you just discussed, that can carry a lethal molecule to kill the prostate cancer cell. That molecule could be radiation. That molecule could be, as you pointed out, some type of an immune cell. But this is a pretty good target for us to do both diagnostics and therapeutics. And this is sort of what we're looking at in terms of discussing this class of drugs. So this is the drug that I'm going to be talking a little bit about. It's got a little bit of an awkward designation right now, so we call it lutetium PSMA 617. And let's just break that down for just a second. PSMA 617 is the targeting agent. It's a small molecule, which is represented over here. And what it does is it's injected into the patient. It circulates around the bloodstream because it's injected into a vein. And then it attaches to PSMA. PSMA is represented by this little blue figure right over here. Or you can see it bigger over here. So here's the PSMA molecule, And then PSMA-617 hooks onto that PSMA molecule. And in this case, PSMA-617 is carrying on its back a payload of radiation. And that radiation source is called lutetium-177. It's a naturally occurring chemical. It's a type of an element. It's found in the periodic table of chemistry. And lutetium emits a certain kind of radiation that's very, very specific. It's called a beta particle, and it emits enough radiation to kill the prostate cancer cell that it's attached to and a few cells around it as well. And so it doesn't emit enough radiation to hurt the rest of the body very much, and it's just enough to kill a cell that it's attached to and several surrounding cells as well. So this is what the drug looks like. It's PSMA-617, the targeting agent, plus, as you just discussed, the linker, and then the payload over here of lutetium. And what happens to it when it hooks up to the prostate cancer cell, the prostate cancer cell takes the whole drug in, all of these parts, into the interior of the cell. And once it's inside of the cell, a process that we call endocytosis, but is basically a description of how the cell brings that in. It just internalizes the whole drug, brings it in, and then the radiation damages the cell's DNA and those next to it, and then the cell dies. That's how this drug works. What we were also talking about before in terms of diagnostics, this lutetium here is enough radiation to kill the prostate cancer cell. But we could as easily, instead of having lutetium on the back end of the targeting agent, have another kind of radiation that emits only enough radiation not to kill the prostate cancer cell, but to take a really good picture of it. That's called a PSMA PET scan. I'm going to show you a PSMA PET scan here for just a moment. So this is what a PSMA PET scan looks like. Over here in the middle, let's just start over here. This is a picture, an MRI of the patient's prostate. This is a little hard to understand, but we're looking at the patient's prostate right here. And this circle over here, where you see these the green lines drawn, this circle over here, this is the prostate over here. This is the rectum over here. And there's a cancer in the prostate here that's a Gleason 9 cancer. And in this patient, he had a standard bone scan, which you're looking at over here. This is a standard way of taking pictures of prostate cancer to see whether it's spread to the bones. And this looks like a pretty normal bone scan. This is what a bone scan looks like in most men. But when you do the PSMA scan, which you can see here, this is a kind of a PSMA scan called an F18 PYL scan. You can see all these little black dots. These black dots represent prostate cancer that's inside of the patient, generally speaking here in the bones. You could not see these on the standard bone scan. You can only see them on the PSMA scan, on the PET scan. And that's because of standard bone scan reveals cancer only where it's damaged bone. But because you have the PSA molecule on the prostate cancer cell itself, you can now image not on the basis of damaged bone, but on the basis of actually visualizing the prostate cancer cell itself, just as long as it has PSMA on the surface. And this is the same scan looking at the patient in cross-section, and these orange dots are the cancer cells in the bone. This is how we usually look at these PET scans in cross-section. It's sort of like slicing the patient with like a pineapple, looking at it slice by slice. But the computer can represent for us where the cancer is in the patient's bones, and otherwise this would be essentially undetectable. This specific kind of PET scan is now FDA approved nationally, and it's approved for scanning men with early high-risk disease and also relapsed disease. And so I think that all of the viewers of this webcast should know that there's this new imaging technology that's now available and just was FDA approved So this is the second PSMA scan to be approved in the US. The first is a gallium-68 PSMA scan. And it's something that your patients, whether they have early high-risk disease or relapse disease, should ask their doctors about. This is an outline of what the VISION study was looking at. So the VISION study is a clinical trial. So it's an experimental process by which patients who had metastatic disease, so disease that already had spread to the bones and other organs, were being tested to see whether or not the lutetium PSMA-617, this drug we were talking about before, can either prolong life or whether it can delay progression of disease. So either one of those two very important things from a patient perspective, delaying disease progression or prolonging life. And the way that it worked is that patients had to have progressed on standard testosterone-lowering agents. That's usually the first maneuver for metastatic disease, plus one of the newer hormonal therapies, drugs that are added on to those testosterone-lowering agents. Generally, those drugs are enzalutamide or abiraterone, sometimes apalutamide, plus chemotherapy, either one or two types of chemotherapy. So these were quite advanced patients. They had progressed through not only most of the contemporary hormonal regimens, but also standard chemotherapy. And so these patients essentially had few treatment options available to them as the next step. These patients, generally speaking, were facing some tall odds in terms of just being able to control their disease. So the way that the trial worked is their doctors picked the next treatment that they would go on to, or ordinarily, that is without the availability of the lutetium PSMA. And that could be anything that the doctor felt was appropriate, but it couldn't be more chemotherapy. Most of these patients had progressed through chemotherapy. It couldn't be some types of immunotherapy. It couldn't be other radiopharmaceuticals, because we didn't have safety data on combining lutetium with those drugs. And then those patients also had to have a positive PSMA PET scan of the nature that I just showed you. PSMA PET, in this case of the VISION trial, it had to be a gallium-68 PSMA, but they had to have disease that showed that they had the target. And then they were randomly assigned on a 2-to-1 basis to get the lutetium PSMA-617 in addition to their standard of care or the standard of care alone. And then they were followed up. And these were the endpoints of the study. We were either looking at something called radiographic progression-free survival, which is the time from when you enter the study till you progress on scans, or if the patient died. And we also looked at overall survival, how long the patient lived. These were the key endpoints. If either of these were positive, we would call this trial a success. There were some other endpoints as well: response on scans, time to a bone event, safety, tolerability, looking at other biomarkers. That was the essence of the trial's design. I'm going to stop sharing now so that everyone can see me a little bit bigger and talk a little bit about what the results were. So in terms of the longevity of patients, so I just want to remind the viewers that these patients were towards the end of their lives. And since they didn't have very much in the way of other therapies that they could go on, this drug, that is the lutetium PSMA-617, added to their standard of care, improved the length of time that they lived. Basically, it was a 38% reduction in the risk of dying that they were able to enjoy as a result of getting the lutetium. And that improved their overall survival by about 4 months. These patients had less than a year to live. And so that 4 months represented about a 48% improvement. So that was pretty significant for these patients. Of course, that was the median. So we do have patients who have really responded for a very long time. I have patients who are over a year out now and still haven't relapsed. The way that this drug is given is it's given every 6 weeks. It's given as an outpatient, and it's given for a total of 6 doses. So I have patients, for example, who received their 6 doses and still, finished up over a year ago, still haven't relapsed. So there can be some significant responders to this. But even for these patients who had a relatively short time to live, it really reduced their risk of dying of their prostate cancer by a significant amount. And in terms of delaying progression, again, these patients were progressing pretty quickly before they went on treatment. On average, most of the patients who got the standard of care progressed after 3 or 4 months. It improved time to progression by another 5 months, which represented a 60% improvement in the risk of progressing. And again, you have around that some really outstanding performers as well. In terms of the PSA response, how many patients enjoyed a PSA decline that was 50% or more from their baseline? About half the patients had a significant PSA response getting lutetium. Less than 10% had a significant PSA response by the standard of care alone. And in terms of complete responses, seeing the scans clear, about 10% had the scans completely clear as a result of treatment with lutetium, as opposed to 0% with who received the standard of care alone. And around 42% of patients had a significant improvement on their scans, as opposed to 3% who got the standard of care alone. So the drug has significant activity in patients who otherwise would really have few treatment options. It prolonged life, and it delayed progression of disease. So we think that this is a pretty important introduction of this drug class, which is the first for prostate cancer of this drug class, in introducing a new therapy for men who have very few other choices very late in the disease. It's reasonable to ask then, well, what are the side effects of that treatment? How's the quality of life of those patients? And in actuality, this really doesn't have significant side effects. So it will cause some dry mouth, a low-grade dry mouth in around 40% of patients. Not so much that it significantly interferes with their quality of life, but enough for them to notice it if we ask them about it. So some dry mouth is there for around 40%. And around 40% during the infusion will have some nausea. A few of those patients will have some vomiting as well. And so we do give pre-medication in order to prevent that from happening. But otherwise, if you compare this drug to some other drugs like chemotherapy for these late patients, this really is a very favorable set of side effects to have. The most significant side effects that we did see were of the hematologic nature, so the blood counts. Around 13% of patients had some significant anemia, might have needed some blood transfusions. And there was also some significant lowering of the platelets, might have needed some platelet transfusions as well. The patients generally do feel good on this treatment, and it is an outpatient treatment, and it's given once every 6 weeks. So I think that overall is a relatively easy way to control your disease, and sometimes durably if you do have this very advanced form of prostate cancer. Keith, I think you're muted. There we go.
Thank you. That was excellent. Dr. Morris, we went over some key points. What I'll do now is I may ask maybe 1 or 2 questions, and then we'll transition over to Dr. Brogan, and then we'll let him present, and then we'll come together and answer questions from those in attendance.
Sure.
One of the questions that I had for you was, I think I wanted people to recognize the average person doesn't understand the difference between radiation and all they know is radiation, my hair is falling out. When we're talking about, we're not talking about gamma radiation here. We're talking about beta radiation. So could you help the audience understand even the difference between the two and how that impacts the quality of life?
Sure thing. So first of all, there are a number of different kinds of radiation. And so the one word radiation covers several different categories. The first thing is that when most people think of radiation, they're thinking of something called external beam radiation. And that's when the radiation source is outside of your body, being passed through your body. Usually you're on a table in a machine. That's some of the viewers of the webcast might have experienced external beam radiation when they were getting their primary prostate cancer treated at their first diagnosis, because it's a frequently used technique for controlling disease in the prostate itself. And that kind of radiation is quite different from the kind of radiation that I'm talking about here. This kind of radiation isn't delivered through a machine from the outside of your body. It's delivered from the inside of your body because it's an injectable liquid form of radiation that can circulate around the bloodstream. And so this isn't like a beam that's passed through your body from the outside. This emits a tiny amount of radiation that only can go a very small distance and irradiates the prostate cancer cell from within the prostate cancer cell. Mm-hmm. That's what makes beta radiation unique and separate from the kind of radiation that most people are thinking of when they think of radiation therapy.
Okay. Well, thank you. I'm going to put you on hold for a second, and I'm going to bring in, I'll let Dr. Gronin speak, and then we'll come back together and we'll answer questions, because I want you to think about high-risk disease, how you define high-risk disease and who falls in that category is one of the questions that I'll ask.
All right.
Thank you again, Dr. Morris.
My pleasure.
Dr. Gronin.
All right.
Hi, how you doing, sir?
Great,
Yeah.
Great. Appreciate that.
Yeah.
Thanks again for the invite and the opportunity to speak with everybody. And, you know, as I start off, I just wanted to kind of touch on a couple of things you mentioned in your introduction. And, you know, when you made the comments to the, you know, the folks attending, when it comes to your disease and being patient with these new therapies coming on, you know, when I first met Mike Morris, it was in 2014 when I was at Endosite, and we were working on a PSMA-targeted chemotherapy. You know, and when I think of like the evolution of even just the PSMA-targeted therapies that are out there, I mean, we were working on this chemo payload that you're talking about. And then I was involved with the acquisition or the in-licensing of PSMA-617 by Endosite. And that was kind of like the next evolution where it was, you know, PSMA-targeted radiotherapy. And then, you know, when I moved from Novartis to Amgen, now it's PSMA-targeted BiTE therapy. So, you know, it's a very rich environment. Like you said, there's a lot of emerging technologies and targets. So, you know, your main focus today is PSMA, but there are other, you know, when you mentioned the theranostics part, that was the second thing I was going to mention. There are other targets that are, you know, validated and being explored. You know, STEAP1, for example, is another one that Amgen is working on. DLL3, for those that when they get even more progressed disease, you know, they can get into this neuroendocrine-like setting. So DLL3 targeted therapies are now things that are being explored. So there's a lot of opportunities out there, you know, so for those that have prostate cancer, you know, there's options out there that are being either emerging or the ones that are just approved. So, you know, Mike talked about what was just recently presented at ASCO and inevitably will be approved here shortly. And the thing I'm going to talk about today is the PSMA targeted bispecific compound that Amgen is working on. And again, this is one that's kind of in the category of immuno-oncology. So I'm going to present just a couple slides to talk about, you know, what is unique about this platform and, you know, what differentiates it from other immune therapies you might be familiar with. So hold on a second, let me share my screen. All right, let me put in presentation mode, make it a little bit easier. Okay, so first of all, when we talk about BITE technology, that's an acronym that was, kind of coined by Amgen. And what that stands for, it's a bispecific T-cell engager. And when you think of T-cell technology, oftentimes that's kind of an autologous type of therapy where they have to harvest the T-cells from the patient, do some type of manipulation, and then reintroduce those to the patient for them to work. And the way that would work really is if you look at the left side of this panel, that's illustrating what would be kind of a normal immune recognition system. And if you envision the purple cell is basically the cancer cell, and then the blue cell will be the T cell. And your normal immune recognition system basically involves what is considered, when you look at the MHC, that's a major histocompatibility complex. It's kind of a cluster that's on that cancer cell that provides some type of handle or some point of recognition for your immune system. And then when you introduce the T cell to that, it has its TCR, or the T cell receptor, binds with this MHC complex, and that's how you have that recognition of something foreign in your body, and your immune system attacks it. And then that'll go through this process of attacking that that cancer cell. However, there are some cancers out there that are considered cold in the sense that they don't have this normal immune recognition system enabled. So what we have to do with prostate cancer, because that's one of these things that's considered a cold cancer, is what can we do to kind of bring that T cell in close proximity, like Keith had mentioned, in order to have this, this immune response occur where we can fight the cancer cell with our own immune system. So essentially what the BITE technology is, it's a way of kind of bringing those two together. And the way that's accomplished on this slide, what that illustrates is on the left side, you have a monoclonal antibody. So that's a biologic compound that's synthesized that'll recognize a very specific antigen with a very high affinity. And what you can do is you'll take just that binding domain of that monoclonal antibody. And then if you look at the middle cartoon representation here of the BITE molecule, that purple end of the BITE molecule is that recognition end on that tumor-associated antigen. And you can think of this as kind of a modular platform in the sense that depending on what the target is we're going after on that cold tumor, we can modify the construct of this BITE molecule. Like I said, in the case of what we're talking about tonight, this is PSMA. So we're looking at a PSMA being the tumor-associated antigen that we're designing here. And then on the right side, this is the recognition side for the T cell. So if you see the little inverted Y on the right side, that's a monoclonal antibody that is designed to recognize the CD3 component of the T cell, or on the surface of the T cell. So if you just take that binding moiety on the end of that monoclonal antibody, remove that and put that together, you make a smaller biologic that is now this, this engager molecule. So its job is basically to bring recognition on one end of that molecule to the T cell, while it's carrying along with it the cancer cell that has either PSMA on its surface, DLL3, or STEAP1. So this is something that Amgen has been working on in the space of prostate cancer, but they're also the lead company in this type of technology of bispecifics. So in 2014, there was an approval of a drug called Blincyto for ALL. And that was the first bispecific T-cell engager that was approved. So it's a proven platform in that regard. And Amgen is really just looking at how we can look at other targets, other indications, other disease types where we can use this technology in order to help out patients. So I'll stop sharing just for a moment and show basically, let me find my next thing I want to share. All right, I'm not having luck finding that right now. But what I was going to show you was the clinicaltrial.gov posting of the AMG 160 clinical trial. So where we're at right now with the AMG 160 clinical trial, it's in phase 1 still. So in phase 1, what the objective there really is assuring patient safety. We just want to make sure that this technology works. Like I said, there's an approved drug in ALL, but not in prostate cancer that Amgen is working on, but we're testing this technology in prostate cancer. So the main objective really is looking for safety of this technology in prostate cancer patients. We're also looking at what is called the recommended phase 2 dose. And what that is we want to basically increase the dose to a point to where you have the maximum benefit or response with that therapy. And then from there, we'll move into a registration goal of having a phase 3 study, similar to what Mike just presented on vision. So we want to have a phase 3 study using AMG 160 that can provide some results in a positive way for patients to where we can look for approval where this can now be broadly available for patients. So that's where we are with AMG 160 right now. It's still in phase 1. We're doing the safety assessment. We're looking at ways to make this as, is least burdensome for patients. Again, since this is a immuno-oncology type of platform, there are certain side effects that need to be managed that are unique to immuno-oncology. Like Mike mentioned when he went through all the potential side effects of the PSMA 617, each molecule does have its own nuances of things that are needed for management. He mentioned, the dry mouth. With AMG 160, for example, there's a phenomenon called the cytokine release syndrome. And that's just something that needs to be understood and managed. So in phase 1, we're also collecting that safety information so we can inform the physicians on how they can manage that in order to, again, help the patients as much as possible without really impacting their quality of life in a way that they need any hospitalization or any other side effects that might, hinder, that place they're at in their journey with the disease. So happy to answer any questions with regard to AMG 160, the BITE technology in general, and immuno-oncology as it relates to prostate cancer. Thanks, Keith.
Yeah. Thank you again, Mike. That was excellent. And one of the questions that I wanted to ask is, it's a 2-step process. So how are you using? How are you detecting PSMA on the surface of the cells? Are you using the gallium 68 or as a means of screening, or how do you detect it? Because it seems like there's a nice overlap between the diagnostic aspect, and once you know that your patient is expressing high levels of PSMA, and especially in your AMG 160 trial, that this person may be a good candidate. Were you using those type of technologies, or did you just screen for just metastatic castration resistant?
So that's a great question. At least with the phase 1, we've been scanning everybody with PSMA PET. So right now it has been with gallium 68, but there are other technologies out there like Mike had mentioned. So PYL is another option. And, this is an interesting space, right? Because with the radioligand therapy that Mike had mentioned, the common acceptance is that you need 1,000 electrons delivered to the cell to induce, apoptosis or killing that cell. When you're talking about immuno-oncology and the introduction of a T cell, do you need that many receptors on the cancer cell? And we don't know if that's the case.
Yeah.
So, the rules that are applied to, one type of mechanism of action with the PSMA therapies don't necessarily apply equally across the board. So although we are doing that in this phase 1 study in order to understand, the utility of this PSMA PET as it relates to patient selection, that may not require the same type of assessment, where there's a specific rule set applied to say if a patient is eligible.
Oh, so now
Right.
I understand what you're saying. So in this case, the level of PSMA on the surface of a cancer cell, prostate cancer cell, may be lower, but because you may be using, in this case, T cells are using granzymes, so you're using your CD8 T cells, cytotoxic T cells to kill the cancer, then you may be in a position to be a little bit more sensitive or have broader coverage.
Correct.
Yes, so that's interesting.
Yeah, so I mean, and that's always the question. I mean, to what extent do you need, expression of PSMA in order to have, a pharmacological effect? And we don't know, depending on the mechanism of action. So all we can do is collect the data, see what it tells us, and then go from there. And the other thing, and I can't recall if Mike mentioned this in the study, was that if 87% of the patients have PSMA positive disease or they're eligible for the study, at that level of prevalence, do you really need to select patients in the first place? So that's, these are all questions to be debated and, and answered.
Yeah, because one of the questions one of the questions I had that I left with Mike was high risk patients. How are you defining a high risk? Because in African American men are classified as high risk. So that's almost saying that your therapy or Mike's therapy, every African American man should be at least being undergoing some type of PET, CT, or gallium screening. Now I'm going to bring Mike back on. Mike, Morris, Dr. Morris, could you come back on? And
yep, coming right back. Oh,
then that way
you need to turn my video on from your end. There we go. All
okay,
right.
Thank you.
Very good.
Thank you. So what I wanted to do is one, there was the issue of now that you have these clinical trials. I know, Dr. Gronin, that your group, the Amgen 160 is in phase 1, and you're still progressing. Dr. Morris, your clinical trial isn't recruiting any more patients. So the question that I asked, to me, and especially a large percentage of the audience that we have are African American men and their families. African American men are at high risk for prostate cancer. And if that's the case, and NCCN classifies them as high risk, should not, African American men with advanced disease or African American men that have recurrence of biochemical recurrence, should they be first in line, or should they be offered the opportunity for this type of diagnostic tool?
Sure. So I think that we would generally say that anybody who has high-grade disease, Gleason 8, 9, or 10, or Gleason 4+3, or has a very elevated PSA, or has locally advanced disease, so they have a prostate cancer that's involving most of the prostate and perhaps beginning to move out of the prostate. Those patients certainly should get a PSMA scan if they can. It's not available across the country yet,
right?
Since the approval is pretty recent, but it will be disseminating. Now, those are the classic definitions of high risk. How the issue of the African American population should modulate low risk and bump low risk patients up into high risk, and thereby be eligible by virtue of strictly just being African American. I don't think we have the answer to that. And I don't want everyone to think that if they have a low PSA, low Gleason score, and a small primary, that they need a PSMA scan, because there are established diagnostics for that group of men. And we just don't know if getting a PSMA scan for low risk disease otherwise, and someone who has all those features of low risk disease, but who is African American, would benefit from getting a PSMA scan. That's a question that we have to answer, but we just don't know that answer at this juncture. So many of those men, remember, will require no treatment, right? A low risk prostate cancer patient doesn't necessarily need an intervention at all, much less an additional diagnostic study. But how the African American patient will benefit from a PSMA scan if they don't otherwise have high risk or intermediate high risk disease, it's a question we just don't know yet and the answer to.
We have a few questions from the audience that I would like to ask our panelists. The first one is PSMA PET been ordered on a routine basis for patients with early diagnosis with a Gleason 8 and a very high PSA?
So right now, no, because PSMA PET has been, in the US at least, so restricted because it's been experimental. The first PSMA approval was back at the turn of the new year in like January. And that was only at 2 centers in California. The approval a couple of weeks ago was the first commercial national approval. So it's not been routinely done for anybody. And this is really the first window of access that the whole US has. But certainly in the clinical trials that have been conducted, that's the kind of patient that was eligible, the one with the Gleason 8 prostate cancer, yes. But not routinely, just on studies. But now it's a different day and age.
Thank you. Yes, Dr. Morris, what is the status of the VISION trial? Is that trial completely closed at this stage for patients?
Yeah, the trial is done, right? So that's how we know the data. Everyone's been accrued. Accrual is now closed. But what the important question is there access to lutetium PSMA? And if not, how will access happen, right? So there are a couple of steps that have to happen. First, there is a program for compassionate use that the company is running, although patients do need to get a PSMA scan somehow. So these technologies are both rolling out at the same time. So there is going to be a brief period in which it's going to be hard to get that match of both a PSMA scan and access to the drug. But the company is applying for FDA approval, and the FDA itself has indicated that this is a drug that they're going to prioritize the decision-making around, because I think that the agency itself does see the importance of this drug. And especially because the patients that that the VISION trial specifically examined have so few treatment options. So I think that process will move along pretty quickly. But we're not talking about general access to the drug from an approval standpoint until another several months. And then after FDA approval, there has to be a production ramp up in which the drug is made and then shipped. And then the centers themselves, the cancer centers, probably will need to build out from some facilities, because this is a new class of drug for prostate cancer, and it is radioactive. So it has to be given with certain licensing and certain types of rooms that are shielded from the rest of the patients. So there's gonna have to be some logistical changes to the cancer centers and cancer docs in collaboration with nuclear medicine and radiation oncologists to make facilities to give this kind of treatment. So there's going to be a stepwise process. Look, if a person lives in an area that has, a wealth of resources, nuclear medicine, major cancer centers already, then that last piece isn't so important. But still, it's going to be a process to get it to everybody who needs it.
The concept of compassionate use is an important one for patients that are, let's say, metastatic, and they may be running out of options at this stage. So where does that process stand right now, Dr. Morris? I know you mentioned that request is in.
Yeah, so there are a few different programs that patients might be accessed, might have access to. I think the best way to do that is to talk to your oncologist, because the information to get into those programs is through the medical system. But the company itself does have a program in which patients can get lutetium. And then there are separate protocols that cancer centers themselves can open to enable patients to get access at those cancer centers as well. Those are far and few between, though. And so I think that the best thing to do is talk to your own oncologist if you're a patient in this circumstance, see which program is most feasible for that oncologist. But it's, you know, the data just came out a few weeks ago, and it's going to take a little while for the operations around these programs to catch up to the patient need for it. But the best way to access it is patient's own oncologist.
Yeah, I know we did have one comment in our Q&A box that one patient's oncologist indicated that it was not available on compassionate use. And so that, I think that's what you're saying, it's a process. And I would imagine to those patients out there that those centers who are involved in the clinical trial would be your best opportunity to find a compassionate use when it is available. So you can, you know, go to clinicaltrials.gov and in the VISION trial and get a list of those centers that participated in the clinical trial. And I would say we will make that list available tomorrow on our website. We just copy that list of the centers that participated in the VISION clinical trials so that you will have that information.
Yeah, Tom, and the same thing that a lot of people, there were some questions about people who were interested in clinical trials. The Amgen's AMG 509, which is the STEEP clinical trial, and the PSMA clinical trial, Amgen 160, we have already hosted on our clinical trial website. So if people are interested in some of these cutting edge next generation clinical trials, we've already in our Find Your Trial tool, and then some of those clinical trials are hosted or posted on our site. So I just want everybody to know there was a question about can they find information about the clinical trials, and I just wanted everybody to know that we have it out there. And
let me follow up with that because our mission really is at PHEN is to address and eventually eliminate the African American prostate cancer racial disparity. And we preach all the time that we have to be involved in clinical trials for this to happen. We need to be in front of these new developments so we understand how they work for African American patients. So we want to again take this opportunity to encourage those patients to look at these trials, the PSMA trials, the Amgen trials that are now open, and see whether there are opportunities for you to participate. Dr. March, do you know how the VISION trial, the percentage of African American patients that were accrued to the VISION trial?
Yeah, it was too low. It was less than 10%.
Okay.
And I think that everyone involved in that study is acutely aware that there needs to be dedicated studies for the African American patient population for radioligand therapy. There are specific barriers to this as well as a drug class, which I think are really important for the community to focus on overcoming. So not only do you need to get access to radioligand therapy and live near a cancer center that has that multidisciplinary care, because it's not given by the medical oncologist, it's given by somebody licensed to handle radiation, which is either nuclear medicine or radiation oncology. But you also need a PSMA PET scan, right? So there's two significant barriers there. And I think that in addition to all of the systematic barriers, you have a difficult treatment because it's requiring a special study which isn't readily available, and it's requiring multidisciplinary care, and it's requiring a center that has the capacity to do radioligand therapy. I think that this is something that is a really important deficiency right now, and a set of barriers specifically for the African American population that must be focused on and must be overcome. This is something that the company is very aware of, that the academic community is very aware of, and through my participation in the healthcare policy community, that on a policy level is very aware of, and the ability to get radioligand therapy out to the African American community and start hammering away at those barriers is very much appreciated and is the subject of a lot of intense focus right now.
Thank you. I have another comment question here. I have a PSA of 29, and I am scheduled for PSMA and PET next week at Memorial Sloan Kettering in New York. So, Dr. Morris, this is a patient you may see next week.
Or maybe already has.
Oh, maybe already has.
That's great. I'm glad to hear that he's getting what he needs in terms of imaging. That's terrific.
Great. Let's see, do we have any? I do have a question from whether the presenter's slides will be made available. Let me say that this whole presentation will be available on PHEN TV very soon. I was going to say within next week, but I just realized that we're coming up on the 4th of July, and I've gotten a vacation notice from the person responsible. But you can look for this. You can look for this presentation to be made available on PHEN's website very soon, and probably before the end of this week. So so look for this, and you will have a chance to review all the slides, and plus all of the rich discussion that's taking place.
And don't forget, if you have a question about clinical trials, you can always call us too. So we're online, and then we're here for you.
Okay, let's see, do we have any additional questions that we have not addressed? Well, we have one, but I think we've answered that question.
Yeah.
And whether the drug that Dr. Morris spoke about is available for men that have localized prostate cancer. And has this drug come to market, or is it just available in clinical trial? And I think we've answered that. It's coming to market with the FDA approval based upon the VISION trial. And then we hope patients will have some access to compassionate use prior to that. How can a man with prostate cancer find information about enrolling in the clinical trial? I will echo what Dr. Crawford mentioned before. This trial and the trials at Amgen are conducting on our website. So you can go to our website, phentrials.com, and you will get all information that you need for enrolling into the trials that Dr. Gronin spoke about.
One thing that your questioner asked there is, would a patient with localized disease be a candidate for the lutetium treatment? And the answer to that is no, because it has only been tested in men with metastatic disease. And when it does get its FDA approval, we anticipate that it will mirror the same late treatment group that the VISION trial focused on. But there are clinical trials looking at this treatment for men with earlier disease. So there's one that's opening, or actually it's open for men who have not yet received chemotherapy. Remember that the VISION was for men who have already relapsed through chemotherapy. And there's another trial that's opening for newly diagnosed men with metastatic disease who have not yet started their treatment even with hormonal therapy. So there are a couple of earlier trials that are open. There aren't right now in the US studies looking at men who don't have metastatic disease, but I do suspect that the field will be going in that direction, because that's the general direction that drug development goes in the United States. It starts out late and then moves into earlier and earlier treatment groups. And there are some studies overseas looking at men, for example, who are going to get a prostatectomy getting this drug. So as a combination for men with localized disease. So I think that eventually you'll see this technology, if not this drug itself, applied for earlier patients. I think that that's
there's a lot of good
going.
There's a lot of good results coming out of Australia.
Yes, indeed. Australia has a significant radioligand therapy community of investigators. They have a very, not only a lot of dedicated resources to radioligand therapy, but a very loose or looser than we have a regulatory environment that allows them to use these drugs in contexts somewhat earlier than we can.
I want to, you mentioned some of these new developments, and I want to let our audience know that in September, Prostate Cancer Awareness Month, PHEN will be hosting the 17th annual African American Prostate Cancer Disparity Summit. And it will be, we will have sessions over 4 days, September the 8th, 15th, 22nd, and the 29th. We have a part of our agenda will focus on new developments. This certainly would be an area that we will focus on, and we will bring you more information about PSMA, some of the trials and some of the developments that are taking place. So please put that those dates on your calendars. We will be sending out information, but on Prostate Cancer Awareness Month, our 17th annual African American Prostate Cancer Disparities Summit. I have another, Dr. Morris, you have some patients online. I have an appointment with Dr. Morris next month. Can I get the PSMA PET scan prior to my appointment?
Very good. Well, I look forward to seeing you next month.
Okay. Well, this is, I think those are the questions that we have at this point.
I had just one, I had one question about PARP inhibitors, Dr. Morris, and the use of lutetium. I think that could either further enhance the response, or the response is just that great that you don't really need to add an additional treatment option.
Sure thing. So just to explain to the audience, the PARP inhibitors are a class of drugs that work quite well in a subgroup of men with prostate cancer who have biology in their disease that interferes with the repair of DNA. And that's about 20 to 30% of prostate cancer patients. Well, I should say maybe probably more than 10 to 20%. So it's not a huge group of men. The drugs work well in those men, but most men with prostate cancer don't have those mutations that interfere with DNA repair. So the big question is would a PARP inhibitor work better in a man with normal DNA repair if he were receiving a DNA damaging agent like lutetium? And could then PARP inhibitors work in the general prostate cancer population as opposed to specifically the population with a DNA repair deficiency? We don't know the answer to that, but it's a really provocative thought that you could take this class of drug, which right now only works in a small fraction of prostate cancer patients, and give it with a radioligand therapy and have it work in the rest of the 80 to 90% of the patient population. So it's a question that's intrigued me and many of my colleagues. There are some early combination studies looking at this, but to answer that question, we're going to need to first find out if these two treatments can be safely combined with some safety and dosing studies, and then we'll move on to the efficacy studies. But it's a really good question.
Okay, thank you very much.
Sure.
Thank you both.
Yeah, we want to thank you both for taking the time to be with us today. The information shared was very enlightening. I learned a lot, and I'm sure our audience learned a lot. So as I said, this is a hot topic, and it's one that we're going to continue to stay on top of, and I expect to see you all with more information at our upcoming summit. For our audience, thank you so much for joining us. I hope you certainly benefit and learn from the program tonight, and tune back in to our next webinar and put on your calendar our 17th annual African American Prostate Cancer Disparity Summit. I want to remind you that we will submit to you, once we close this, a poll, and we would appreciate your completing that poll. It's very simple, very quick, won't take more than 5 minutes, but that information will be very helpful to us and shaping our education program. So with that, I would like to say thank everybody and good night, and enjoy the 4th of July. Good night.
Good night.
I thank you for having us.
Yeah, thank you.
Thank you. Thank you.
Take care.
Okay. Bye.
Bye-bye.